Salt, it's an essential part of life. It improves the taste of our food, it helps with nerve signal transmission and muscle contractions in our body. It even keeps our roads safe in wintry weather. But what happens when salt washes into our rivers, lakes, and soil? How safe is it for the environment and our friendly plants? Let's explore as if we're in your AP Environmental Science class. Every organism, from tiny microbes to large humans, has a range of tolerance for environmental conditions such as pH, temperature, and salinity. Within this range, they can survive and thrive, but if conditions get too extreme and move outside of that tolerance zone, it can cause stress and even death. Scientists can figure out an organism's tolerance range by first identifying the lethal dose 50, or LD50. LD50 is the concentration that kills half of a test population, and its name comes from L for lethal, D for dose or concentration, and 50 is half of the test population. Today we'll use radish seeds to explore the model of salt toxicity. And here's everything we're gonna need to do that experiment today. We need our test population, and that's gonna be our 50 radish seeds. We're gonna need some petri dishes and some paper towels. And that together is going to serve as our ecosystem to give the perfect environment to allow or not our radish seeds to germinate. Next, to make our salt solutions, we're going to need some water, some salt, a beaker, and a scale to measure out our 3% salt solution. Finally, once the germination has occurred, we're going to need a ruler and some miscellaneous lab equipment to get things done. So let's get started. The first thing we'll do is make our stock solution that is 3% salt. And to do that, I'm going to use our scale to weigh out 3% or 3 grams of salt. So I'll weigh out 3 grams of salt, and I'm going to dissolve that in a total of about 97 milliliter of distilled water. The salt takes a little bit of room, so that's why I'm using 97 instead of the 100. We'll add our distilled water to that, and then we'll stir the salt in until it's fully dissolved. Once the 3% stock solution is ready, subsequent dilutions can be made to get salt solutions with lower concentration. But to do that, it's going to require a little math. To do this math, we need to use our dilution equation. And remember, the dilution equation is the concentration of our initial solution times the volume of our initial solution is going to equal the concentration of our final solution times its volume. And because we know some of these parameters, and because we're starting with a 3% stock, we can start completing this equation. The concentration of our final solution we're trying to make is 2.5% salt. So let's start filling it out. We know the concentration of our initial, that's 3%. We're trying to ultimately figure out how much volume of our initial we need to make our final volume. We're trying to get to 2.5% salt, and we want 10 milliliters of that final 2.5 concentration salt. So now we're trying to figure out VI, and so to do this, we need to work some of this equation around so that VI is going to equal 2.5% times 10 ml divided by 3%. And so with a little math, we can figure out that we need about 8.3 milliliters of our 3% stock solution, and we're going to queue it up to 10 mLs with 1.7 milliliters of water. And if we do that at the end, we're going to have a concentration of 2.5 salt with 10 ml of that solution. So now that we have our 2.5% salt solution, we need to make all other 8 solutions so that we can actually do the lab. Instead of sitting here and watching me do all of these boring math calculations, why don't you have a try? That should have been enough time for you to figure out what that math was. But in case you haven't, here's the amount of 3% stock solutions and water you'll need to make the dilutions needed for the lab. Now that we figured out our different dilution tables, let's actually make these solutions. You'll need your stock solutions and some conical vials, and of course, water to make that. Let's go. We have our salt solutions. Now it's time to set up the actual experiment. So to do this, we'll need our Petri dishes. And so we'll take that and we'll put on one of our cut paper towels. You just need a single layer and it needs to fit relatively well at the bottom. And you need to start with labeling these so that they don't get confused whenever you have 10 different Petri dishes. So remember to label both the top and the bottom so that in case the lids come off, you always can figure out what goes with what. So we're gonna start off with our 0% salt or just water. So to do this, like I said, you'll place your paper towel at the bottom. So you'll grab your 0% or just water and you will barely moisten it using this transfer pipette. So to do this, you'll probably need about 2 milliliters of water, but it can vary depending on the type of paper towels that you have. So as shown, you just want to get everything wet. You don't want to get everything dripping. So don't oversaturate your paper towels. Once that paper towel has been moistened with the correct solution, that's when we're going to add 10 radish seeds in your Petri dishes. So you want to do these kind of nice and even. So I'm going to lay out kind of a nice pattern where I do 2, 3, and then 2, and another 3. You want these to be relatively well spaced and not crowded in one area. So once that's completed, you'll just go ahead and close these up, and we'll make all 10 other Petri dishes as well. So now that we've got our Petri dishes set up, we're gonna put these in a nice dark place and give it 3 to 4 days to allow our seeds to germinate, or maybe not, depending on the salt that is present in their different ecosystems. When we come back after 3 days or so, we're gonna take the seeds, we're gonna look at the percent mortalities, and we're gonna take a ruler and measure how much they've grown in that time. So stay tuned and come back to see us. So welcome back. It's been 3 days, and what a transformation. The first thing we need to do is look at each of these Petri dishes to see how many have grown, how many have germinated, and to figure out the length of the sprouts in each of these different conditions. So let's look at these. What is considered germination? Well, if you can see here, our 3%, for example, none of the seeds have anything plant resembling except the seed. This is considered not germinated at all. That's very different compared to the 0.01% salt, where you can see the plants are literally growing out of the Petri dishes. So once these little seedlings have left the coat of the seed, then they're considered germinated. So let's count these to figure out what exactly has germinated, what hasn't, and what is our LD50. So now let's look at each one of these and record our findings. So we'll start first at our 0%, and so we're gonna look first at our percent mortality. And to figure out percent mortality, we first need to figure out which seeds germinated and which did not. If we look carefully at these, we can see that all of them have germinated except for 3 of them. And so for this, we considered about 70% viable and 30% has mortality, considering that we started with 10 seeds. So next, our 0.01 When looking at carefully, we can see sure there's some seeds, but these seeds are all cracked open, and if we count the number of actually seedlings we get, we see that 9 have germinated while only 1 has not. And so you might be asking yourself, Dr. Simon, why does 0.01 have more germination than our 0%? And the reason for that, I believe, is because when creating this experiment, I used distilled water or highly purified water to do this. So as we'll see, maybe some salt is needed, as we know, right? Salt is part of life, but maybe too much salt is toxic. So maybe in this 0% condition, there just wasn't enough salt present. So we had 3 that were not germinated, and we had just 1 in this one. So let's keep going in our 0.05. So in here, we actually have 2 that are not germinated. And so that puts at about 20% mortality. So a little bit increasing mortality as we increased our salt concentration, but let's see if that trend continues. So in this one, similarly to our 0.01%, our 1.1% only had 1 that did not germinate. In our 0.5, we had 2 that did not germinate, or about 20% mortality. So here again, we have all of them germinated except 2 of them. So that mortality is staying equal between the 0.5 and the 1%. So here with the 1.5% salt, you can clearly start to see a big difference in our quality of our germination. Sure, a lot of them germinated in our 1%, 1.5%, but you can just see that the root length is not as long. And that's because salt, sure, it's toxic and causes these to die under the right conditions, but it also stuns their growth. And that's why maybe we can expect this to be a little bit shorter in terms of their root length than some of our other ones with our lower salt concentrations. Now looking at 2% salt, you can see small amounts have germinated, but overall I would say about 80% mortality in this one. So quite a big jump when only 2 out of the 10 germinated. And then our 2.5 and our 3% salts, these look pretty bleak. There's a little bit of a sprout in this one, but it's really not considered germinated because of how small that sprout is. And that was in our 2.5. In our 3% salt, we have 100% mortality. None of them survived that 3% stock solution that we treated them with. So now that we know the overall trend in mortality of these different conditions, let's look at the actual growth that happened or lack thereof. And so to do that, we're gonna need a ruler, and I'm gonna use tweezers to kind of grab the sprouts out of here to figure out exactly in millimeters the length of those sprouts. If you have, like our 3% stock solutions, none that sprouted, then you would count these as 0 millimeters of growth because obviously no growth happened. So if we were to take 3 of these randomly and average them together, it would be 0 millimeter growth for 3%. Our 2.5%, we had just a baby sprout, but that's not measurable by any means with our ruler. So for this one, we're gonna take an average sprout length of also 0 millimeters. Our 2% had a little bit of sprouting going on. So I'm gonna try to take that seed and measure. So looking carefully, I would say we had a sprout length of this one of 4 millimeters. And for the other ones, since we're just taking 3, we'll take 2 other ones at 0 millimeter to get our overall average. So for a 1.5 condition, we had some sprouted and some that were not. So we can take some of these kind of longer sprouts and figure out the overall length. For this one, it is about 11 millimeters for our first reading. Our second one, I think if we were to straighten that out a little bit, we'd get a reading of about 12 or 13. We'll say 13 millimeters. And because we had so many that did not sprout, let's just take one at random that didn't sprout. So we'll take a 0 for that. And so if you want to be super accurate, then obviously you would want to measure all 10 of these. But because I know you don't want to sit here and watch me measure all 10 of these sprouts, just take an average of 3 to get your average. And you can see the grade variation between these. Even though it's the same conditions, one grew significant more than the other. And that's why it's so important to take multiple readings and get that average when you're doing a real life experiment. So now that we have all of our measurements, let's discuss the results and what conclusions this experiment can draw. When looking at our control, we had the growth of about 60 millimeter, which was significantly less than our 0.01%, which had about 90 millimeters of growth. That being said, the reason I think that happened is because while some salt is highly toxic, like we saw with our 3% stock solution, maybe not enough salt is also not good, which highlights the importance of salt both in the environment and in your diet. So remember, some salt is probably okay, but not too much. At the end of the day, what we're talking about today is salt toxicity, and the best way to do that is by looking at the LD50. Remember, the LD50 is the amount or dosage of a certain chemical that kills 50% of your test population. In our case, the LD50 is most likely closest to about 1.5% because only about 50% or so of the seeds germinated. Compare that to our 3%, which had 0 germination, we can see that of course the higher concentration of salt you go, the more toxic it becomes both to the mortality as well as to the growth. And that's it. Today we used math, data, and hand-on experimentation to simulate how scientists determine the toxicity of certain substances. Lessons like these are what you'll encounter in your APES class. There's plenty more in our apes course too. Use it as a companion for your class to prepare for quizzes, tests, and get ahead of your peers in exam prep. Thank you for joining us today. If this video sparked your curiosity, please consider liking, sharing, and subscribing. Your support helps us bring more science to life. Until our next experiment, stay curious and keep exploring.